Reminder, histology is the study of tissues. And tissues are defined as groups of cells with a specific function. So for today's lecture, we're going to look at the major types and subtypes of tissue, their structures and their functions, and we're going to focus particularly on relating tissue structure to tissue function. So for today's study guide, you should know what the major characteristics of epithelium are, what the major characteristics of connective tissue are, the names, structures, and functions of all the tissues discussed. You should be able to visually identify all the tissues discussed. For this, you should use your lab manual, and the posted histology slide handout. You should know the 3 types of muscle, which include skeletal, cardiac, and smooth, and be able to distinguish one from another. And you should be able to draw a neuron and the 2 types of cells in the nervous system. So we have 4 classes of tissues that make up the human body. On the outermost surface, and the innermost surface of all of our organs, we have epithelium. Epithelium is your linings and coverings. Connective tissue connects, protects, and transports muscle and nervous tissue are our 2 electrically excitable tissues. Muscle is electrically excitable and it can shorten and produce force. Nervous tissue is also electrically excitable but it conducts signals from point A to point B. So let's start with our linings and coverings. Let's start with our epithelium. Epithelium has 5 primary characteristics. The first is cellularity. When you're looking at epithelium, what you're going to see is primarily cells. without any extracellular fibers. All epithelium is avascular. It has no blood supply. Which means it depends on diffusion to get oxygen and nutrients and to get rid of wastes. It has a basement membrane. This is a connective tissue membrane. That's located deep to the cellular layer. It has polarity, meaning it has a clear apical surface and a basal surface. And it has a high capacity for regeneration. Now, epithelium is primarily classified based on two characteristics. The first is the shape of the cells. And there are three major shape types. If your cells are thin and flat, this is what's known as squamous or squamous epithelium. If your cells are as approximately as tall as they are wide, this is cuboidal epithelium. And if the cells are taller, than they are wide, this is columnar epithelium. So in order to determine the name of the epithelium, you should look at the shape of the cells at the apical surface. The other characteristic that we use to name epithelium is the number of layers. If it's only one cell layer thick, it's simple epithelium. if it's more than one cell layer thick, then it's stratified epithelium. So this brings us to our first in-class assignment question. Which of the following are characteristics of epithelium? Select all that apply. So the 4 characteristics of epithelium listed here are cellularity, the presence of a basement membrane or basal lamina, polarity, and regeneration. Vascularity is incorrect because epithelium is avascular. So in this slide here, we have 2 forms of simple epithelium. There's simple cuboidal epithelium, and simple squamous epithelium. Our simple cuboidal epithelium makes up our kidney tubules. So this slide is actually taken from the kidneys. And the kidneys are made up of structures called tubules. And so here you can see one of those tubules cut in cross section. You can see the cells of these tubules from our basement membrane are a single cell layer thick, and the cells are approximately cube-shaped. So this is simple cuboidal epithelium. Also found within the kidneys, you have capillary beds and structures that surround the capillary beds called the renal capsule. This renal capsule is made up of a single layer of thin, flat cells called simple squamous epithelium. Now you tend to find simple cuboidal and simple squamous epithelium in areas where you want to maximize diffusion. And you're maximizing diffusion by decreasing the diffusion distance. So this brings us to our second in-class assignment. Blank is the process that will be optimized by having a single thin layer of flat cells. And the answer here is diffusion. A. Our next type of tissue is stratified squamous epithelium. The basement membrane for stratified squamous epithelium is down here, outlining it here in blue, at least part of it. So the epithelium goes from this basement membrane to the surface. We can clearly see that there are multiple layers of cells. Which gives us our stratified portion of the name. And if you look at the surface of these cells, they're flattened, which gives us the squamous portion of our name. This type of tissue is found in the epidermis of the skin. The back of the throat, and around the anus. And these multiple layers of cells provide resistance to abrasion. Because you can easily lose some of the superficial layers when you rub up against a surface or when a piece of food scratches the back of your throat, for example. Stratified cuboidal epithelium. This is only found primarily in two major places in the body. In sebaceous glands, which are oil glands found in the skin, and in the thyroid gland. And you can see here, this is your basement membrane. This is the top or apical surface. So the epithelial layer goes from the basement membrane to the apical surface. There's more than one cell layer, so that gives us our stratified part of our name. And the cells are roughly cube-shaped. They're approximately as tall as they are wide. So this is stratified cuboidal epithelium. Transitional epithelium is one of our unique forms of epithelium. It's found in the urinary bladder and the epididymis, of the male reproductive system. And when these hollow organs are empty, the superficial cells have this distinctive dome shape, and there are multiple layers of cells. But as the organ fills, the cells become more flat in appearance. And there are fewer layers. So it transitions both in between the shape of the cells and the number of layers, which is where this tissue gets its name. This allows for the bladder to fill significantly without a change in internal pressure. simple columnar epithelium is found in the lining of the small intestine. And again, we can see our basement membrane here. So this is our apical surface. Our cells are taller than they are wide. And there's only one layer. These cells, you can see the border actually looks kind of fuzzy. There are microvilli all over the surface of this cell, which increases the surface area for nutrient diffusion. And transport. Another unusual type of epithelium is called pseudostratified ciliated columnar epithelium. This is found in the trachea and in the proximal portion of the epididymis. Remember the prefix pseudo from Bio 105? Pseudo means false. So pseudostratified means false stratification. Cilia refers to the cilia that are coming up from the surface of the cell. And columnar refers to the shape of the cell. So it looks like there are multiple layers of cells here because you have several different groups of nuclei at different levels. But in reality, every single one of these cells has part of its cell membrane that comes down and touches this basement membrane here. So it looks stratified, but it's actually not stratified, which is where the pseudostratified portion of the name comes from. In the trachea, this makes up the mucociliary escalator. So you can see in this slide of the trachea over here, and to a lesser extent over here, part of a cell called a goblet cell. Goblet cells produce mucus. And that mucus is secreted onto the surface of the cells surrounding the cilia. So all the particulate matter, the dirt and the dander and the dust, gets stuck in the mucus those cilia beat and push out of the lungs. and you swallow that mucus that's filled with all of that particulate matter, as well as dead and dying immune system cells, and it ends up in your digestive system. So this allows the lungs to be very, very close to sterile by the time you reach the alveolar ducts and the alveoli of the lungs. It's not completely sterile, but it's close. Stratified columnar epithelium is found in the salivary gland ducts, so the parotid, the sublingual, and the submandibular salivary glands. So the cells look approximately columnar in shape. There are multiple layers of them, and so you get stratified columnar epithelium. So this brings us to our in-class assignment. The alveoli of the lungs would have what type of epithelium? And just remember, the alveoli is where gas exchange occurs. So the alveoli of the lungs have simple squamous epithelium in them to maximize diffusion. The epidermis would have what type of epithelium? The epidermis has stratified squamous epithelium, and this primarily serves for protection. And resistance to abrasion. This brings us to our next classification of tissues, our connective tissues. Connective tissues actually have a whole host of different functions throughout the body. They connect epithelium to the rest of the body. Things like the basement membrane, they provide structure, bone, cartilage, tendons, and ligaments. Are all forms of connective tissue. Adipose stores energy in the form of lipid. And blood, which is a form of fluid connective tissue, transports material throughout the body via the cardiovascular system. So we can classify connective tissue primarily based on its structure and organization. So loose connective tissue, these tend to be relatively disorganized. There's not a lot of consistent internal structure, and there are 3 types in adults: areolar, connective tissue, adipose, and reticular connective tissue. And all 3 of these are kind of the packing peanuts of the body. They fit into empty spaces. Dense connective tissues tend to be tightly packed and more highly organized. So if we look at areolar connective tissue, this is the least specialized and it's the most disorganized. It has all 3 fiber types found in connective tissue. So there are elastic fibers, collagen fibers, and reticular fibers. The cells that make these fibers are called fibroblasts. And outside of your fibroblasts, in addition to the extracellular fibers, you have a thin, viscous ground substance. This is a fluid that makes the tissue slippery. And this type of tissue, when you look at it under a microscope, is never going to look identical in between slides. It's so disorganized that you're still going to see the same materials. You're going to see the fibroblasts. You're going to see the 3 fiber types. You're going to see the ground substance. But how they're put together is kind of random. This apologies for that. This tissue fills the space between the skin and muscle. Adipose tissue is found deep to the skin, and it is one of the few forms of connective tissue that's primarily cellular. The cells in adipose tissue are called adipocytes, and when you look at them under a microscope, the thing you could most easily confuse this with is simple squamous epithelium. But as you can see, each one of these cells here isn't attached to a basement membrane. It's basically a nucleus pushed way over to the side of the cell and a large lipid-filled vacuole. Adipose tissue is your long-term fuel storage. It provides padding, protection, and insulation. There are a couple of different types of adipose tissue. There's white fat, which is the most common form. It is pretty close to avascular, and it's designed to store lipid. It's not designed to be metabolically active. Versus brown fat, which is a current area of research, is much more vascularized. The adipocytes have many more mitochondria, and it's thought this is primarily used for heat and metabolism, because it's much more metabolically active. Reticular tissue is our third form of loose connective tissue. It is a supportive framework for softer organs. Things like the liver, the kidneys, the spleen. It has reticular fibers in it. And it pretty much acts as the skeleton or forming the shape of those softer organs that don't have actual bones associated with them. Dense connective tissue, or sometimes known as connective tissue proper, is going to be tightly packed with high numbers of either collagen fibers or elastic fibers. And it's going to be much more highly organized compared to our loose forms of connective tissue. So if we look at probably the most organized form of dense regular connective tissue, it is forming tendons and ligaments. And it plays a large role as part of the skeletal system in attachment and stabilization. So in terms of attachment, tendons connect muscle to bone, and ligaments connect bone to bone. And ligaments are involved in stabilizing or limiting joint movement. And if you look at dense regular connective tissue under a microscope, what you're going to see is a few fibroblasts, but it's primarily going to be densely packed collagen fibers, all running in the same orientation. Which provides a substantial amount of strength in the direction that the fibers are oriented, but they're susceptible to damage if a force is applied perpendicular to the orientation of the fibers. Dense irregular connective tissue has collagen fibers, but collagen fibers are going in all three planes. So they're in the X plane, they're in the Y plane, and they're in the Z plane. And this provides for strength in multiple directions. So you end up more with an interwoven network of collagen fibers. You find this in the dermis of the skin, particularly the reticular layer of the dermis. Elastic tissue is going to have the similar fibroblast, but instead of having collagen fibers, it's going to have elastic fibers. And for example, you find this making up more elastic ligaments, like the ones between the spinal vertebrae. These allow for more stretch. And possibly energy storage. During locomotion. Our fluid forms of connective tissue include blood and lymph. We're really only going to talk about blood. Our fluid connective tissues are a watery matrix of dissolved proteins, things like albumin, and our formed elements. And in blood, our formed elements are composed of three different types of things. You have your red blood cells, which carry oxygen. Your white blood cells, which have immune functions. And platelets, which are important for clotting. Now, blood does have a fiber, just like most forms of connective tissue. It's just that the only time you're going to see this fiber is when blood clots. So usually the fiber, which is called fibrinogen, is soluble. But then when the clotting cascade gets activated, thrombin gets released. And thrombin converts fibrinogen to fibrin, which helps form a clot and prevent blood from leaking out. Our supportive connective tissue supports soft tissues and body weight, and this includes our 3 types of cartilage: hyaline cartilage, fibrocartilage, and elastic cartilage. As well as bone, which makes up our skeletal system. So if we're looking at cartilage, cartilage does have an extracellular matrix. It's made up of water as well as proteoglycans. These are derived from chondroitin sulfates, and they help both the water and the proteoglycans help cartilage to be relatively incompressible. Which is good because cartilage in many areas serves as shock absorption. The cells in cartilage are called chondrocytes, and they're found in hollow areas called lacuna. There are 3 types of cartilage. The first is hyaline cartilage. Hyaline cartilage is made up of tightly packed collagen fibers, so tightly packed that you actually don't see the fiber structure. This is found at the end of long bones. And is primarily for shock absorption and connection. Fibrocartilage, which is what you're seeing here, has larger collagen fibers in it, so you're actually able to easily see on a microscope slide the wavy collagen fibers. You're still going to see the chondrocytes in the lacuna, and fibrocartilage is found in your intervertebral discs. And then elastic cartilage, you're going to see thin elastic fibers in the matrix along with the chondrocytes and the lacunae. This is found in the external ear, and the epiglottis. The epiglottis is the piece of tissue that folds over your trachea when you swallow and prevents food and water from going down into your respiratory system. So these elastic fibers allow the cartilage to be more stretchy and to recoil back to its original shape relatively easily. Bone we're going to talk about a lot more in lecture 3, but compact bone in particular, which is what I'm showing here, has a fairly distinguished and easy to identify bullseye shape. So with compact bone, you're going to have a central canal. That's going to be where the blood vessels so the arteries and veins as well as the nerves run. Then you're going to have these layers surrounding the central canal. And these layers are called lamellae, or lamella singular. The cells found in bone are called osteocytes. And similar to our chondrocytes, those are also found in hollow spaces called lacuna. Muscle tissue, it's specialized for contraction. So it's important to remember that muscles pull, they don't push. So muscles are designed to shorten and produce force. And any type of body movement that you can imagine, from your heart beating to the skeletal muscles that are active and lifting up a pencil, to your digestive system pushing food through the alimentary canal. All of that is muscle tissue. And there are 3 types of muscle tissue that are classified based on their striation pattern. So if we're looking to see whether there's a striation pattern or not, we're going to look along the length of the muscle cell. And we're going to look for these lines that go from one edge of the muscle to the other. That's striation. The number of nuclei can vary in between 1 to 2, or multinucleated. Another thing that varies between muscles is nervous system control. Is it somatic, which means voluntary, or is it autonomic, which means involuntary? The 4th characteristic is cell structure. Does the cell run end-to-end parallel? Like you're seeing here? Or do the fibers actually come off and form branching patterns? So skeletal muscle, which is found in all of your voluntary muscles, is striated. That's referring to these thin lines that you're seeing here that are running from side to the one side of the cell to the other. It's under somatic control, so it's voluntary. It's multinucleated because the cells are formed by the fusion of multiple stem cells during development. And it's unbranched or parallel. Cardiac muscle, which is found in the heart only, is also striated but it's under autonomic control. It has 1 to 2 nuclei. And the fibers do branch. What gives it away for me, though, when I'm looking at it on a slide are these lines here. And they can appear white or black. These are called intercalated discs. And these are a combination of gap junctions and desmosomes that hold the muscle cells together, but also allow ions to flow directly from one cell to the next. So they're only found in cardiac muscle, which is what helps us characterize cardiac muscle pretty easily. Smooth muscle is non-striated. Has 1 nucleus. It's under autonomic control. And it's unbranched. And it's found in the walls of hollow organs. So your blood vessels, your digestive system, your ureter, your urethra, the uterus, any hollow organ that produces movement in an involuntary manner is going to be lined with smooth muscle, with the exception of the heart, which is cardiac muscle only. Okay, this brings us to our next in-class assignment. Which category of muscle do the following muscles belong to? The heart, that's cardiac muscle. That's the only place cardiac muscle is found. The muscle inside the wall of the aorta, which is the large blood vessel coming off of the heart, is smooth muscle. The biceps brachii is skeletal muscle. And muscle found within the walls of the trachea is also smooth muscle. Neural tissue, or nervous tissue, is responsible for several things: conducting electrical signals, sensation, so measuring the external environment, processing things, and controlling systems. And there are 2 types of cells found in neural tissue. There are neurons, which are ones that perform electrical communication, and then there are neuroglia, or glial cells. These are our support cells, and they do a whole host of functions, which we'll talk more about when we get into the nervous system later on. And this brings us to our last in-class assignment. So we're going to review this at the beginning of the next lecture, along with that one on osmosis. So please take a stab at this, go through the different types of tissue. Each answer is only used once. And again, we'll discuss it once we get back. For the lab, you should log onto Canvas and pull up the Histology lab handout. It's pictures of all the slides that you're going to need to look at. Using those pictures and your lab manual, work through the lab manual, observe all the tissues, and complete your drawings. So make sure you can identify all the tissues from the pictures. Make sure you know each tissue's function, and where we talked about it, be able to relate a tissue structure to its function. Thank you guys very much for being so patient, and I will see you in a week.